We're going to look at why a neuron's resting membrane potential averages negative 70 millivolts. There's three contributors to this. So just kind of to go over the neuron real quick, we have the dendrites, we have the body or the soma, we have the axon hillock, and then the axon, and then the axon terminals. We have the myelin sheath here. So what the first contributor to the negative 70 that sets up a neuron for action potential, it polarizes the neuron, is a sodium potassium ATPase pump. So anytime you hear pump, there's going to be, it's active transport of 3 sodium atoms out and 2 potassium ions in. So you have 3 positive charges going out, 2 positive charges going in. So in the grand scheme of things, this is going to give you about negative 5 millivolts. So it's a small contributor. But these sodium potassium pumps are really important. They are a big part of our basal metabolic rate because they generate so much ATP to keep this concentration gradient established, with sodium being the extracellular ion that's really high, and then potassium being the intracellular ion that's really high. So this pump is in a lot of cells throughout our body, if not all the cells. So that's one. 2 is another protein embedded in the neuron cell membrane, and it's a leaky potassium channel. So because of this concentration gradient established here, we have a lot of potassium inside the cell. And so if there's a channel that allows it out, it's going to efflux out these leaky potassium channels. And as you have those positive charges leak out of the cell, that leaves phosphates and sulfates and proteins, all these different negatively charged anions inside the cell. And so that's going to contribute the majority, and it's actually going to overshoot. It's going to be a lot more than negative 70 millivolts. And then we have the third contributor, and that's leaky sodium channels. So we have this concentration gradient where sodium wants to go in. What's important to realize here is that this leaky potassium channel is much more permeable than the sodium channel. So you get a lot more potassium leaking out of the cell than you do sodium leaking in. And so say we get negative, if we started at 0 millivolts and we just looked at the sodium potassium pump, we'd be at negative 5 millivolts roughly. And then throw in the leaky potassium channels, maybe we're at negative 90 millivolts. Then the sodium's going to bring some more positives back in, so that might bring us back to negative 70 millivolts. So you can kind of see an idea of how that interplay takes place to give us that average resting membrane potential. In the next video, we're going to talk about hitting threshold, which is negative 55 millivolts. And then that sets off an all or none phenomenon of an action potential. So we'll get to that in the next video.